Hi, my name is Dr. Dan Altura. I have a BS, an MS, and PhD degrees from the UCLA School of Engineering and Applied Science. I've been involved in research for over 40 years. My specialties are chemical kinetics, electrochemistry, and also material science. What I want to quickly discuss today, you can get a better picture of what's going on by viewing my video, which takes about 25 to 30 minutes. I'm going to try to condense this in this particular video. We're going to discuss Darwin's tree of life. Basically, Darwin says in his book that he published in 1859 that life, all the species we have today, including human being, were produced by a random, by chance process. And this is called evolution. And the way it's done, little steps in each generation. Little steps, very many little steps over a long period of time. We start with a single ancestor cell, and we go to all the different species that are alive today to simplify this. And this is what I learned when in my freshman year at the engineering school. A lot of times when you have a very complex system, if you want to simplify it, if you can simplify it, you can get some information which you can then apply to the complex system. So we're going to simplify this. First of all, we're going to just start with a single cell. There's a question mark here. We're not going to know what happened in Darwin's tree of life. We're just going to go from the single cell to the human cell. Now, the human cell is also too complicated. The human being is too complicated because the human being consists of 37 trillion cells. A number that I can't even imagine. And also the human cell itself is too complicated because each human cell consists of 20,000 different proteins which have different jobs. So we're just going to look at two different proteins, BRCA1 and BRCA2. These are tumor suppressor proteins. If these are mutated, this causes an increase in the rate of cancer for ovarian cancer, breast cancer in women, and also some cancers in men. Very important proteins and the coding genes that produce these proteins. Now, so we're just going to look at these two proteins. Now, what single cell are we going to use? What kind of model? Well, I won't get into it here. You look at my other video. But basically, we're going to use a model of the simplest bacteria out there, where they actually took this bacteria called M. and reduced the number of proteins down to the simplest level where if you knocked out one more protein, it wouldn't function anymore. So that's going to be the basis of our single cell. And because this cell is so simple, and this cell does not suffer from cancer, a single cell doesn't suffer from cancer, we're going to assume that BRCA1 and BRCA2, these two genes, and the proteins they produce don't exist in a single cell, but do exist in a human cell, which we know. One thing about these proteins, like any proteins, the proteins have two major ways they function. They have to have, the geometry of the protein has to be exact, and the electronic configuration of the protein has to be exact for the proteins to be able to function properly. And this is similar to an immobilizer key in a car. The key has a geometric component to it, but also it has a little integrated circuit in there which has an electronic code which corresponds to an electronic code in the steering wheel. So this is very important. Each protein has a geometric and electronic component which makes it very specific. If one of these amino acids, proteins are made of amino acids, it's a long chain of amino acids. If one of these amino acids is missing or is being replaced by a different amino acid, etc., these two things get messed up, and the protein cannot work properly. BRCA1, these two proteins are what are called tumor suppressors. They work in the cell to prevent cancer, basically. They're very large proteins. One is 1,863 amino acids long. The other one is 3,418 amino acids long. These again are tumor suppressors, and they're multifunctional, which means they don't only do one tumor suppressor job, they do several. And new jobs that these two proteins do are found all the time. Every few months they find another job. They're involved, for example, in detecting mutations in the DNA. They're also involved in and double-strand repair. In other words, if the strands of DNA have a mutation, for example, BRCA2 can be used to repair and replace that part of the double strand. They're involved in stopping the cell cycle. Cell cycle is when the cell divides and goes into two cells. If the DNA is defective, these two proteins are involved in stopping the division process so the cells cannot divide and turn into a tumor. Finally, they're also involved in what's called apoptosis. What is that? If the cell DNA cannot be repaired, then the cell is basically killed off. You kill off the cell to save the human being. These are totally unique, both from each other and also from every other protein in the human cell. You can't just take bits and pieces of different proteins in the cell and produce either one of these. They're totally unique, which is very important. They weren't copies or anything. And if they're mutated, we can see that they produce, if BRCA is mutated, for example, they've discovered over 1,700 mutations that cause 850 clinical significant diseases. 850 different forms of these two cancers. Finally, going back to the immobilizer, both of these have what's called a Goldilocks component to them. Each one of these large proteins can only be just right. The combination of amino acids, which is coded by the gene for each of these, has to be just right. You can't even have one amino acid missing or replaced by a different amino acid for these things to work right. There's only one combination. So how many different combinations are there? Because that will tell us how to find, how many different combinations it's going to take to get to each one of these. So we take an example of a necklace, 4 positions on the necklace, and 5 different types of pearls. 5 pearls, 4 positions. The number of different combinations is you take the 5 pearls of the one type, multiply by the 5 pearls of the second type, the 5 pearls of the fourth type, third type, and 5 pearls of multiply by the 5 pearls of the fifth type, of the fourth type. You get a total of 625 pearls, 625 different combinations. The general formula for these combinations is T to the P, where T is the number of types and P is the total positions. In these large proteins, the amino acids that you have is basically you have 20 different amino acids, and the combinations are basically using this formula. You have 20 to the 1863 power, a very large number. And for BRCA2, it's even larger, 20 to the 1818 number. These combinations are so large, at 3,400 zeros, if I pronounce 1 zero every half second, it would take me over a half hour to pronounce all the zeros. Now, how do these compare to other examples? Well, Powerball, the different combinations are about 1 billion, 10 to the 9th, very small. The total number of atoms in the universe. Trillions of galaxies, billions and billions of stars in each galaxy. For the total atoms in the universe, you have 10 to the 80 total atoms, and you have 10 to the 86 total particles in the universe. Imagine trying to find one single neutron in the protein, in the universe, trying to fly around at the speed of light in this universe, which is billions of light years wide, trying to find this one special neutron that will be the Goldilocks neutron. This is basically impossible to do. It would take a tremendous amount of generations, billions and billions times billions of generations, and billions times billions times billions of years. Much older than the age of the Earth, and much older than the age of the universe. And the path in getting from a single cell to the human cell is important. But we see here, it's not important. Left off a T over here. The path is not important. We can go in any way we want from a single cell, which doesn't have Darwin's, the BRCA1 and BRCA2, to the human being which has both of these. So we see that the path is not important. The path is not important. So the odds we have here, because we have all these combinations, apply no matter which evolutionary path you go through here. And by chance, the chance of getting the right combination is basically nil. Even smaller proteins, like the average protein in a living in a single cell, which is about 280 amino acids long, that also has 280 zeros compared to the 80 zeros of total atoms in the universe. Also impossible. And also, if you take the largest protein in the body, titin, which is it's called titin, it's a protein used in human muscle. This protein has 34,000 different amino acids. They all have to be strung together in the proper way to make them effective. Now, so the odds, if you look at the video that I have, a larger video, whether it's Powerball or PC or roulette, to generate these random numbers in Powerball, they're all the same. The odds are really the same. And the same thing is true here for a single cell, and going from a single cell to a human cell, where you don't have BRCA, and here you do have BRCA1 and 2. So the odds are minimally small, but again, it would take billions and billions of generations and billions times billions of years to get this, to have enough time to produce either one of these two proteins by chance. And this is not some intellectual exercise. You have to understand, we're talking here about saving many thousands of human lives. Whether it's intelligent design or evolution, it's not a theoretical argument here. We're talking about, you have to have the right perspective in doing research and finding a solution to this problem. Perspective is very important. I found that over many, many years of practice in my own field. Thank you very much.